All right. Welcome, everyone. We'll go ahead and get started. So welcome to the NIH Director's Lecture. This is a very special lecture, and it features leading researchers from around the globe. Nominations are taken from across NIH, and typically only about 3 NIH Directors' Lecturers are chosen each year, so this is a big honor. And it is our honor to welcome Dr. Eddie Chang today to spend this time with us. I am personally very, very grateful. Eddie is so well known to us. I was thinking on the way over here that in neuroscience circles, anyway, at NIH, all you have to do is say Eddie, and everyone knows who you are. So that's kind of like Madonna to us. So before I introduce Dr. Chang, just a few housekeeping items. So if you're attending this lecture for CME credits, the code, as you can see there, is 57680. If you are watching online and you'd like to submit questions, please click the link for Send Live Feedback, and your question will be relayed to us, and we'll present that to the speaker at the end of the lecture. And you can submit questions at any time during the lecture. So Dr. Chang is the Joan and Sanford Weill Chair and the Jeannie Robertson Distinguished Professor of Neurological Surgery at the University of California in San Francisco. His clinical expertise is in surgical therapies for epilepsy, pain, and brain tumors. He specializes in advanced neurophysiologic brain mapping methods, including awake speech and motor mapping, to safely perform neurosurgical procedures in the eloquent areas of the brain. So Dr. Chang is really the consummate surgeon scientist. He's a practicing surgeon, he chairs the department, and he does all of this incredible research that you'll hear about shortly. His lab has demonstrated the detailed functional organization of the human speech cortex and has translated these discoveries toward the development of a speech neuroprosthetic device to restore communication for people living with paralysis and speech disorders. He directs or co-directs the Center for Neural Engineering and Prostheses, and that's a collaborative enterprise of UCSF and UC Berkeley. The center brings together experts in engineering, neurology, and neurosurgery to develop state-of-the-art biomedical technologies to restore function for patients with neurological disabilities, such as paralysis and speech disorders. So today, he'll describe critical advances over the past decade that have been made by his lab in deciphering the cortical signals that underlie our ability to hear and speak words. He's the author of hundreds of papers. He's the recipient of many awards, and he was elected to the National Academy of Medicine in 2020. I am so excited to welcome Dr. Chang today and look forward to your talk.
Thanks, Dr. Tucci. That was really a generous introduction, and I also want to thank Dr. Shore for the invitation to be here. It's an absolute honor and privilege to be at the NIH. I mean that really from the deepest part of my heart. And I want to first start by thanking all of you for the work that you do, and in particular for the collaboration that we've had over the last 2 decades in our work. We're just going to switch the slides here. Go ahead. Okay, so the focus of this lecture is really to just focus on a part of what my lab studies. We study both how the brain processes what we hear, and then also how we create speech. And I'm going to focus on the latter, the process of the cortical control of speaking, and how we've used that knowledge to build a translational device for people who have lost the ability to communicate. And so one perspective that is really important to understanding how we speak is the fundamental process of speaking is about how we shape the breath. Speech starts with the exhalation of air from the lungs. When it gets the level of your vocal folds, it starts the vibrations there. and that is the voice energy of our words. That voice energy then goes up through the upper part of the vocal tract, the lips, the jaw, the tongue, where the movements create a filter on that sound, and that's what gives rise to all of the consonants and vowels that we speak. So a lot of what I'm going to be focusing on is the control of the vocal tract. How does the brain allow us to control that ability to shape the breath? And it's really a remarkable process, one that's unique to our species, to create words, and something quite specialized. When I started this work, it was about 15 years ago, started in my lab, it became immediately obvious to me that we did not understand very much about the organization or the basic principles by which the human brain processes words. And so in addition to studying how we hear, we decided to do studies that were going to be directly related to how we produce and articulate. And 99% of motor neuroscience is really focused on the arms and limbs, the arm and hand, right? The vast majority. But how much we actually study the central control of speaking is very understudied. So this image is actually an illustration from Harvey Cushing, who's considered the father of modern neurosurgery. And one of my residents gave me this illustration, which hangs on my wall in the lab. He was describing one of his patients that actually was shot in the head with a bullet entry up here in the forehead, and then developed seizures from part of the temporal lobe before down here where there were seizures. Now, the reason I'm showing this is not related to his injury, but if you look closely to this, it's really one of the first descriptions of the somatotopic or body organization of the part of the lateral motor cortex that we call the homunculus. Now, this was later popularized by Wilder Penfield. And this is something we have all known, right, even from undergraduate psychology and neuroscience classes. And so what he writes here is that there's a part of our motor cortex that corresponds to movement of the face, corner of the lips, the jaw, the tongue, the palate, the back of our throat, and then the vocal folds. So basically there was a map in all of our brains that recapitulates the spatial organization from front to back of the vocal tract. And so that was the starting point. So no one had ever described how this works while we actually speak. This was always in the context of stimulating these areas and seeing movements. And so I became very interested to understand how to relate these two phenomena in the quarter in understanding speech. So the main technique that we used was a technique, and we still use, it's called electrocorticography. These are electrode arrays that are placed on the brain of patients with epilepsy. When we started this research, we increased the density of the arrays, which lay right on the brain surface. This allowed us to ask deeper questions about the neural coding of these areas without introducing any additional risk. More electrodes, but just on the brain surface, meant more data. And this is an example of a 16 by 16, 256 channel array. And still, this is the most highly resolved method that we have in human neuroscience, both in terms of spatial and simultaneous temporal resolution to understand the processes that are happening in the cortex in real time. So in this movie, you'll basically see the activation of the pre- and post-central gyrus when someone is speaking a sentence.
Shipbuilding is a most fascinating process.
So that pattern of activity is what we refer to as the neural code. And what we have deeply, deeply focused on for the last 15 years is trying to understand how that neural code works. How does electrical activity that's reflected in what you just saw in that particular part of the brain, how does that correspond to different aspects of speech control? And so I'm going to talk a little bit about how we've deciphered that code, what are the things that we've learned from it, and then how do we leverage that code for something translational. So the first study that we did was extremely simple. In 2013, we published this study where we had participants just reading a list of syllables. Ba, bu, pi, ra, yi. Kind of boring, right? Very, very, very simple. And turned out it was really the first time that it was done in this kind of resolution. And if you look at these three different syllables, like ba, da, ga, when we're producing these sounds, when you make that ba sound, your lips come to closure. It's the release of your lips that creates the ba sound. It's the tongue tip to the top of your upper tooth. That's called an alveolar in linguistics. That creates the da sound in dad. And the ga sound is the back of the tongue going up. Okay, that's quite incredible. When I learned all of this, I was really astounded. I had no idea how I was actually speaking. But this is something that we all learn without any explicit knowledge, which is really quite incredible. But nonetheless, this is the way that we produce these sounds. And if you look at the different electrodes that are sorted from top to bottom here, these are the same set of electrodes across these three different utterances. You can see that they have overlapping but also distinct patterns of activation. So for example, in this ba sound, the B sound, you see these electrodes that are activated here, but not so much in the D and the ga sound. Whereas in the da sound, this electrode is activated, but not in the others. This one as well. This ga sound has an activation down here that's not in the others. And then there's some that are just overlapping across all of these. So it's not as simple as like this one area is one syllable, etc. There's a complex kind of representation that has distributed information. So one way that we took to try to understand this was to take all of those different sounds. For example, all of the lip sounds like the ba, fa, ma, pa, those are what we call labials. And we were able to correlate those with the electrodes across patients that were correlated with that kind of movement. And so what this is basically a plot, a spatial plot, sorted from top to bottom, front to back. And the electrodes centered around here, around the central sulcus, were the ones that are associated with these labial movements while people were speaking. And then if you look at a different set of articulated sounds for the larynx, we in fact found this one at the very bottom, which I showed you earlier in the Cushing illustration. There's this vocal fold representation down at the homunculus, the motor cortex, somatotopic organization. But then we found an additional laryngeal area that was not described and was very, very hard for us to understand. We did multiple studies to try to make sure that this is real, and I'll describe one of them. But when you look across multiple patients, we see this premotor laryngeal area, lip area, jaw, tongue, larynx, and all of this area is dynamically activated when we speak. This was the first organization, functional organization of the speech motor cortex that was generated from people speaking, and it recapitulated a lot of the things that we knew from before. However, there was also this new thing that we discovered, that there was an additional area of the larynx. One of my students, Ben Dichter, was very interested in this finding, and he actually did his whole PhD on trying to pin down and understand how the larynx, the vocal folds, are controlled by the brain. And so the vocal folds, in addition to sort of like producing the energy of the voice, it's what modulates the pitch of my voice. So when I stress a particular word, that's primarily because I'm changing the pitch of that, and that's by tensing the vocal folds. So what Ben discovered in a paper in Cell in 2018 was that this area in particular, this previously unknown laryngeal motor cortex, is directly responsible for tightening the vocal folds. And so what he did was he did an experiment where people basically said this same sentence over multiple trials, but on a given trial, they stressed one word. And we did these kind of randomly. So in one instance, I never said she stole my money versus I never said she stole my money. You can change a statement to a question just by changing the pitch of the voice. And that's how we use this in English. In tonal languages, it has a different purpose to change directly the word meaning. So what Ben found was basically we found this cluster of electrodes both on the left and right hemisphere that were strongly correlated to the pitch of the voice. very strongly correlated and they were tightly clustered. And it confirmed what we found from our earlier studies using syllables. If you looked at all of the trials and you looked at the pitch tracings of those utterances, these are the ones that are in blue here. And underneath that, we're looking at the single trial neural activity. We look primarily in the high-frequency component that we call high gamma. You can see that there's single trial time-locked responses to what word was stressed. And so when someone is stressing a particular word, the vocal folds are tensing, and those electrodes that correspond to that are in this particular part. So we verified that there is one. Now, Ben also showed that when people are singing melodies, the do, re, mi kind of thing, the neural activity changes there. So it's not something specific to speech. It's actually something more rudimentary like the vocal fold tension. And then something that was unexpected but quite interesting was that when we played back people's speech through a speaker, we found that these areas were also activated, encoding what they heard. So this means that there's auditory information in this part of the brain as well. It's encoding the pitch of what people heard. And so in a separate set of experiments in patients that were undergoing brain mapping as part of their sleep surgeries, what we did was we used endotracheal tube, a breathing tube that my colleagues in ENT use for mapping, the nerves in the neck when they dissect tumors there. But here what we were doing was using this endotracheal tube that has wires that non-invasively contact the vocal folds after the intubation. And we could stimulate in different parts of the cortex to see which parts of the cortex would evoke movement in the vocal folds. And what we found was, again, that there was this cluster of sites in the dorsal laryngeal motor cortex that evoked movement here. So here we could prove that this was a motor area, not just a sensory auditory one, but in fact a motor and sensory. In fact, if you increase the magnitude of the stimulation, you see an increase in magnitude of the EMG. So there is a part of our brain on both sides that controls the vocal folds and in a very direct way, and we think it's a disynaptic connection. What about what's happening above the vocal folds? So the movements of speech are very complex compared to the movements of the limb. You can model the skeletal movements around limbs by their joint 2D, sort of movements. The muscles of the lips, for example, have a totally different set of kinematic properties, more like a sphincter. The tongue has the biomechanics of a hydrostat. So these are very, very complex muscles to understand. Nonetheless, we can measure and study their position using a variety of different tools from vision and ultrasound. But one of the things that we found was that if you look at one particular electrode, these sites in the motor cortex are encoding specific movement trajectories. They are encoding specific movements of our vocal tract when we speak. By themselves, they have no meaning, but when you put them together, they give rise to all consonants and vowels. So this is an example of one particular electrode here, right on the central sulcus, that is very important for encoding the duh sound, this particular coronal trajectory, where the tongue tip goes to the back of the upper tooth, the tongue back goes down, and the jaw goes up and down. So the activity of that one particular electrode is encoded by a movement that you can see in this video that's plotting the trajectories. The tongue tip goes forward, the tongue back goes down, and the jaw opens and closes. That's a coordinated movement across the vocal tract, across a couple of articulators, all encoded at this one particular spot. All of us have this, kind of encoding of this sound. That's how we make the duh sound. If I compare that to another electrode that's just a centimeter away, it's really important for this labial trajectory. And you'll see that the main movements here are around the closure of the lips. So we have these different spots in the motor cortex that encode different trajectories of movements that control the different parts of our vocal tract. Again, by themselves, they have no meaning. But when you sequence them, you put them together, and they're working in a coordinated way, it gives rise to all consonants and vowels, which give rise to all syllables in all words, and from there, all possible meanings. So thinking about these as fundamental building blocks for communication, for speech. So one thing that we can do is we can look at this data across electrodes. Each column here corresponds to a different sensor, and each row corresponds to different parts of the vocal tract. And below that are all of the different phonemes in the English language. And so for those of you who do genomics, it kind of looks like a plot that's a whole genome study. Here it's just showing you all the different electrodes and how they're activated by different productions. And one of the reasons why I show this is that as early as, probably about 8 years ago, we had this intuition that we now could access the entire phonetic alphabet, the electrical coding, a dictionary, so to, in some way, of the electrical patterns for every sound in English. And that was a very powerful point in the lab because we then started to think about translating this kind of technology for people who were paralyzed. So I'm going to talk a little bit about what we've done with that kind of insight to build a speech prosthetic. And this quote from Stephen Hawking is particularly poignant. Although I cannot move and I have to speak through a computer, in my mind I am free. So this encapsulates part of the problem that we're trying to solve for people who have a condition called locked-in, where essentially they have intact cognition, but are not able to send the signals out to the mouth. There's a variety of different conditions like ALS or brainstem stroke that can cause this, and several others. And so we've endeavored to build essentially a device, a technology that can translate that code that I've been describing into something that can directly read out and produce speech that you can hear. And so the basic question that was framed by this MIT tech review that was covering some of this early work was, could a person who is paralyzed and unable to speak, like Stephen Hawking, use a brain implant to carry on a conversation? Okay, so that was the basic question that we wanted to pursue. And we wanted to leverage all that we had learned from that early period about how that code works. Now, one question is, well, why speech? Why not just have people communicate through handwriting, a BCI, brain-computer interface, of what people are trying to express by their handwriting, or what they're trying to type, or what they're trying to text? Those are all effective ways. In fact, the whole field was focused on getting patients who are paralyzed to be able to communicate through moving a computer cursor quickly to different parts of the keyboard. And we wanted to do something different because speech is a different mode of communication altogether. It allows us to communicate at rates at between 120, 150, 200 words per minute. That's why it's such a special behavior for our species. It allows me to transmit information from my mind to yours and back in a very fluent way, allows us to build civilization, allows us as social beings to transmit ideas, to build, to learn, and very, very foundational. It's such an important part of being human. So this is why we really went straight to this ambitious idea of trying to do a speech prosthetic. And early on, a lot of my closest friends and colleagues said, this is impossible. Like, too complex. Speech is too abstract. And I kind of agree with them. But at the same time, we were seeing this electrical code that was laid out in front of us. And so we said, okay, well, why don't we give it a try? So one of the first things that we did was to try this in epilepsy patients as a proof of principle. And these are people who are speaking, normally speaking, and we could record what they were speaking into a microphone. So we could compare what we could decode from their brain activity compared to what they actually said. It was actually a really good way to start because we knew exactly what they said, and we could try to build a computer model that would decode what they were saying just from the brain activity. And so that's what we did in this study that was led by Gopala and Josh in the lab, graduate student postdoc. And in this particular approach, what we did was we translated the brain activity to a virtual simulation of the vocal tract in a computer. And then we took that virtual simulation and had that create words that you could hear. So it was a two-part decoder. One part is translating brain activity into movements, a simulation of the vocal tract, and then translating those simulated movements into sound. And it is possible to go from brain activity to the sounds, but one of the reasons we chose this method is because we knew this part of the brain is controlling movement of the vocal tract. So we wanted to be sort of as biomimetic as possible, match the model to what the brain is trying to do. And this is an example of that speech. Here's a spectrogram of the speech that's recorded by the microphone on top for these two sentences. And then what was generated by our first decoder of those two sentences, just from the neural activity. And you can see that it has a lot of the same energy contours, a lot of the gross spectral and temporal information was there, but a lot of the fine structure of the speech was missing. And so we were excited to see that it had this visual appearance that was close. But what was the most exciting to us was that some of the sentences were actually intelligible. A lot of them were unintelligible, but some of them were actually intelligible, and I'll share some of those with you. But that was a milestone, really just being able to prove to ourselves that it's actually possible with these epilepsy electrodes that were never designed to do this kind of purpose. So in this video, you'll basically see the brain activity for a given sentence. You'll see it controlling a virtual model of the vocal tract that we built. And then you'll see, you'll hear what the model is generating. And then you'll hear what the person actually said to compare.
The proof that you are seeking is available in books. The proof that you are seeking is not available in books. The proof that you are seeking is available in books. Shipbuilding is a most fascinating process. Shipbuilding is a most fascinating process. Shipbuilding is a most fascinating process.
So what was exciting was, for the first time, hearing a voice in this synthesis. It's raspy, it's noisy. And if you hallucinate, you'll hear voices in there. Statistically, it's very much there, but really just at the borderline. But that was exciting to actually hear, and that we could use that with some of the machine learning models. And this is just an example of why we did this with this kinematic model. The MCD just is a coefficient that expresses how similar the original spoken spectrogram is to what is decoded. And if you go straight from brain activity to sound, you can do it if you have enough data. But by building an articulatory model of the vocal tract, it requires much less data. If you're trying to build an AI that goes from brain activity to sound, it's got to learn a very, very complex mapping. And the strategy here was, let's reduce how much the network has to learn by imposing some structure that we know to be true, that the vocal tract movements are a causal mechanism of sound generation, and put that in, and that can really reduce. And reducing the training time is very important for people who are paralyzed, of course, because it can't take forever. Okay, so we basically convinced ourselves that we could try this in someone who's paralyzed. And so what we did was we created the Bravo trial at UCSF, and it stands for the BCI restoration of arm and voice. There's basically two labs, my lab and also Karnesh Ganguly, who's a rehabilitation neurologist who's studying how cortical representations around the hand can be used to control a cursor or a robotic arm. The part that my lab was focusing on was the part of the array that was important for speech. So we have two different parallel but complementary streams, really from one array. And we first started with a 128-channel electrode that was covering these areas that I've described for people who have severe paralysis that was impairing communication and limb movement. So these are very, very severe cases of paralysis. And this was approved with an IDE. So the first participant that underwent the study was described in this paper in the New England Journal. It describes the first successful decoding of words and sentences from someone who was paralyzed. And I'll tell you a little bit about his case. So Pancho was the first person in the world to have this. He's still implanted now about 6.5 years after the original implant. And he suffered a severe stroke about 15 years ago in the brainstem. And the way that he normally communicates is with this stick that's attached to his hat. He can move his neck, and he types things out on this assistive device, which many people use who have communication disorder. It's called AAC. And so the goal here is to allow Pancho to be able to communicate at much faster rates and more. And so this is what his MRI looks like. So this is an important concept. Where his stroke was here in the part of the brain we call the brain stem. And the brain stem is what connects the brain to the spinal cord and all of the cranial nerves. So as you can imagine, he can't get the signals out of his brain because he had this huge stroke that occurred in the pons. But everything that is above that is working normally. So his thoughts, his ideas are all there. He just can't get them out because the communication to the external world has been severed. So what we did was we built an array. We took the arrays that we were using for epilepsy, and we worked with some of the BCI companies, in particular BlackRock, that had this port that had been improved primarily with microelectrode arrays called the Utah array. We said, well, instead of the Utah array, I want you to just attach it to one of these ECOGs because I want to just record from the brain surface. And so we were able to do that, and the FDA was okay with that because after some testing, we could prove that this had the same safety as, the forms that it's already been approved for. So it's taking two approved products, combining them with some additional safety to just prove that this new form was going to be okay. So this is the first chronic ECOG, and the way it basically works is that it records from Pancho's brain. The signals are taken out. They're translated and digitized from analog brainwaves into digital neural signals. The computer is doing signal processing at the first step, picking out the parts of the ECOG that are most informative. The second step is an AI algorithm that's detecting when he's trying to speak. The third part of this algorithm is determining what word he's trying to say. And in this first experiment, it was just among 50 words, so it's giving us the ranked probability of 50 words and then displaying the first one on a screen. So this was really the first attempt at this. And so I do think it's important for people to understand what this is about. This is about a person who has volunteered to be part of an experimental study the first time it was ever done, who now has been living with this port on his head, his percutaneous port. And this is what that connector looks like, and you see an HDMI cable. And you might be able to appreciate it, but this has 128 little gold contacts. Each one of those contacts has a line, an electrical wire, that goes to a different part of his brain. So this is how we're literally tapping in to Pancho's brain using this port. And he's had this for quite some time, but we're moving towards wireless to solve this. So the surgery takes about 4 hours to do this, but where we spend hours, days, months, and years is trying to understand the signal that's generated from it. And the challenge that we had with this is to translate this kind of signal into words. It's in there, but that's really what we do. Any of us who study the brain and the neural code this is essentially what we're trying to do, which is to translate the electrical code into something that's interpretable. And so this is an example of which electrodes on Pancho's brain were the best for detecting when he wanted to speak. It's near that laryngeal motor cortex that we described earlier. The area that's shaded is the entire motor cortex here. And then the areas that were really important for classification were the area down here in the ventral part of this motor cortex and this area in the middle. The first 50 words that we chose to work on showed decent, not perfect, far from perfect decoding accuracy, but it basically worked. We could discriminate different words from one another, and not surprisingly, the words that were similar, like it and is, were pretty confusable. And the words that were phonetically dissimilar were much, much more discriminable. And that should make sense based on all the previous work that we showed about how this information is coded in the part of the brain. We were also able to see over weeks, months, and now years, a stable activation of these electrodes over time, because that was one of the main reasons we chose this approach as opposed to microelectrode. We wanted something that was really clinically stable over some time, to be able to show that that's something that someone could use over a long time period. So this is the demo video from Pancho's. So someone in the lab is going to write to him. You're going to see down here what's decoded from his brain activity. This is the port with his HDMI cable. The blinking dots means that this blinking dots here corresponds to the algorithm that's detected that he's trying to say something. That's the speech detection. And then the rest of the time is trying to figure out what word he said. And once it figures out, it will plot. Now, it's trying to rank all 50 words, and it's just going to show you the top ranked one, but it keeps track of all other 50. Okay, so this was doing word by word. The analysis is trying to chunk it by words and gives us that. This last one I included because it's got an error. It's got a couple of errors, but it uses the autocorrect language model. And because it keeps track of the ranked probability of all the words, it can use the context of the words that were before in order to update either the spelling or the word choice. So we're using a lot of these things that all of you are familiar with from texting and word processing, like autocorrect, in order to accelerate and make it more accurate and whatnot. It's what we call language model. Okay, so when Pancho's paper was published, a woman named Anne reached out to us from Canada. Actually, it turned out she had a similar situation. She had a brainstem stroke about 18 years prior, and she could not move her limbs and could not speak. I'll share with you a video that kind of shows you her vocalizations.
That you are not from here. Tell me about where you're living right now.
So Anne uses eye tracking technology in order for her to communicate. She's an incredible person and raised two incredible daughters, actually, this whole time that she was paralyzed. It's a really incredible story. Brainstem stroke in the similar part of the brain. And because of the results that we saw with Pancho, we went back to the FDA and asked if we could increase the channel count, actually double it from 128 to 253 channels. So more information. And again, this is not in the brain. This is recording just from the brain surface. And we immediately saw much better performance with Anne. One of our internal metrics is having people speak NATO code words like alpha, beta, Charlie. It's a standard task that we can quantify and compare across individuals. And within days, about a week and a half, Anne was able to get between 90 and 100% accuracy with this. And one of the things that we did was we froze the algorithm, the classifier that was analyzing the data at about 2 weeks after we had started all this, and used the same algorithm for weeks and months later, and we still could get really good performance. That's really important. It means the decoder is stable over time and therefore could be practical for someone to do without having to train it every day. So we wanted to do something even more ambitious with Anne. So we built a multimodal speech neuroprosthesis. So instead of just having it decode her brain activity to text, like I showed you with Pancho, we had a separate parallel AI that was translating her brain activity into words that you could hear, called speech synthesis. And then another one that is decoding facial movements associated with speech through an avatar. So 3 different AI engines, all based on speech, but different ways of expressing speech. One is text, one is speech synthesis that you can hear, and the third is expressive, what it looks like. And the reasons why we did this was because we are interested in exploring how having embodiment of speech can help people give feedback in order to learn the articulation. As they're learning to speak again, what's it like to actually have an avatar of yourself and see and understand what's working or what's not working? What we learned from this with Anne and Pancho is that it goes beyond that. Being able to choose the eye color or the hair color, the features of their avatar, actually was restoring some of their identity. So a lot of people who have lost the ability to communicate actually have not just lost their voice, but they've lost, a lot of the personhood and identity. We want it to be more natural, and someday this will be a form that will facilitate virtual social interactions. So this is the model that we built a couple of years ago. It's undergone a lot of change since then. But the basic structure is we use a recurrent neural network that goes from brain activity decoding the different phonetic elements. We use CTC beam search in order to arrange those phonetic elements into word forms. And then we process this into a text so that it can be read out. And then we use a language model in order to clean it up. So this is how we go from brain activity to text. And in this particular thing, we're giving these sentences to Anne. There's a countdown. And we're asking her to just repeat these so we can quantify how accurate she is in articulating these different words and sentences. Now, because we had a lot more data, a lot more information, a lot more resolution, we weren't doing word-by-word decoding with Anne. We actually asked her to speak whole phrases. So it was a lot more ambitious, actually, what we were doing. The blinking dot still means it's detected that she's trying to speak, but then the analysis is now analyzing the whole phrase and sentence, not word by word. And you can see that it is a lot faster. Actually, we got up to about 80 words per minute at about 75% accuracy, so significantly faster with vocabulary around 1,500 words, and which can expand to several thousand. That was super exciting to see, and took several months of doing. Now, what about the speech synthesis? We used a similar neural network to translate the brain activity into units of speech. These are latent units that have been learned from a speech recognition model called Hubert that was produced by Meta. And we were able to translate those into speech that you can hear using a vocoder. And in this particular case, we used Anne's original voice that was from her wedding that we had access to, so we could quasi-personalize it to her. And then built that with an avatar that was also using a similar machine learning method.
I think you are wonderful.
So it's decoding the text, the speech
Give
synthesis
me a few minutes.
and the movements that are associated with those words simultaneously.
What do you think of my artificial voice?
Uncanny valley, right? Kind of strange to see someone communicating in that kind of way. Very surreal, too. Because we built an avatar, I asked the team, well, let's try something outside of speech, just facial expressions. And that works well. You could classify a lot of different facial expressions pretty easily in emotional expressions. Here's just some examples of Anne expressing a fake smile and some other things as well. And, all of these things link back. When we look at, as she is making these attempted speech movements, we can actually go back to the very first principles that we discovered, 10, 15 years ago about this map. The larynx, the hand area, the lips, the tongue, etc. It's all related. It's the same kind of principles that are involved. Alex Silva is a graduate student who wanted to see how this would apply to bilingualism. And because Pancho was actually, when he had his accident, he was actually a monolingual Spanish speaker, and he learned English after his accident. So we went back and built a bilingual decoder. I'm not going to go into the details, but it basically uses some of the similar principles, but it has a language model that's specific to English or Spanish, and only presents the one that it determines it's whether English or Spanish. So it's the same brain area, but it's basically detecting if the sequence of elements corresponds to English or Spanish, and then just displays that one. So that's an example in English. Here's going to be another one in Spanish. We don't tell the decoder, of course, what language he's trying to speak. It has to infer it based on the sequences of the phonetic elements and the brain activity. Okay. Another thing that is not published yet, but I'm sharing it, it's already in press, is from two graduate students, Kayla Littlejohn and Cho et al. But here we're trying to make the synthesis faster. If you noticed in the previous example, there's a long latency between when they're trying to say it and it actually being said. So we've tried to reduce that to about a second, and we're doing real-time synthesis. So here's an example of that.
I am sorry, but I have an appointment now. Let me tell you what I did.
Okay, so that's an example of what we call streaming synthesis. That we can take it out with relatively low latencies, and every month, week, whatever. These are just going optimizations, getting better and better all the time. And what's been exciting is that this field of speech BCI is exploding. Other groups using different approaches are finding incredible results. The BrainGate group using microelectrodes has really astounding results. Other groups as well. And so the New England Journal asked me to write a review, and in this, I put forward some criteria, what we should look for in a device and technology. We want it to be high performance, fast, accurate, something easy to use. We want it to be robust. We don't want to spend hours calibrating this kind of thing every day. We want it to be stable. We want it to be used across a lot of different indications, not just one. We want it to be able to use in different settings, different disease stages. We want it to be private and safe. We want it to be fully implantable. We want this wireless. We don't want the port. That's not scalable. And we want it to be surgically as easy as possible, not only to implant, but also to remove. So this is kind of the wish list, and we're working on that. And here are some of the optimizations. We can predict from the ECOG activity how to get the very high accuracy and speed rates from our papers and others. The projections are actually very similar to what you look at microelectrode. The number of sensors matters. Where the sensors are and their density really matters. But we now have the core set of data to make those kind of projections and do the optimization. So I'm going to summarize here and then get to a little bit about a big picture. So what I've described here is a path that we've taken from the basic science of the cortical physiology of the way that we speak. It goes really deep. I've only shown some of the highlights related to this map of articulatory movements, this laryngeal motor cortex that was really underappreciated, how movements of the vocal tract are encoded by cortical activity in this part of our brain. When you have a stroke or injury there, you lose the ability to articulate fluently. It's what we call dysarthria. We've taken that knowledge to translate it to build now high-performance text synthesis and facial avatar readouts, decoders of that. This is really the era of AI and using that technology. We can embody speech by putting it in virtual forms that can not only enhance verbal but also nonverbal communication. And we know some of the basic parameters we need to optimize. So this is no longer a question is like, can this be done? It's a question now of how good can we make it? It's not about if this is possible. It's about when and what form. And we're very, very focused on that. Okay, so big picture. I think it's very important to me personally, like this invitation to be here today, because none of this would have been possible without the support of the NIH. And I feel personally like extremely lucky to be a benefactor, to collaborate with the NIH at large, in particular the NIDCD, but also the Brain Initiative. I'm just really one of the luckiest people in the world to be able to do this work that's been so fulfilling and productive. I do want to call out Roger Miller, who's here. This was part of his master plan. There's a workshop in 2006. It was the first NIDCD workshop on brain-computer interfaces for speech synthesis. And I remember reading the summary from this. I wasn't even at an eligible age to attend this workshop, but I remember reading the archived summary and thinking, wow, this is super exciting. Is this even possible? And it planted a seed in my mind, like, I want to work on something related to that. We need to know the science. And it took a while to get there. But if you notice that this is 2006, that's almost 20 years ago, Roger. So thank you for the vision and the foresight. Decade and a half later, we were able to get supported by a clinical trial program through NIDCD. Trinley and also Roger support our program. It's the first pilot clinical trial for speech neuroprosthesis. So what I just showed you in this translation was really sponsored by the work of NIDCD and the NIH. And so this timeline is my sort of reflection of what my career and our work has done. And there's many other labs that have this parallel kind of trajectory. This first description of the speech motor cortex was in 2013. This was sponsored by a New Innovator Award from the director's office. It was called Functional Architecture of Speech Motor Cortex. And I was able to double down on that in the NIH Brain Initiative. They gave us all of this information about the larynx cortex, this coding for consonants and vowels. And from that, we were able to do the first speech synthesis that I showed you and do a lot more applying AI. And we're able to then build the evidence and the scientific case for moving to a patient scenario where people who are actually paralyzed. Take this out of the epilepsy monitoring, move it to people who are suffering, and continuing to optimize to get high performance streaming speech synthesis that's now supported by this very special UL1 and then also a new UG3 that is supporting development of the hardware, a wireless system, so that we don't have to deal with this crazy percutaneous port. Super important for us to do the initial de-risking, but now we have to use much better technology. So we're hoping for the first in human fully implantable device in the next 2 years. None of this would have been possible without the support of the NIH or NIDCD and Brain Initiative. The last thing I wanted to describe is where we're going with the science, full circle. Talking about science to translation, back to science. And we're using a new tool called NeuroPixels. It's a high-resolution way to study neuronal activity at single-neuron scale. One of my friends, Karim Zagul, is like one of the world's pioneer of single-unit recordings in human. He's using a slightly different technology that's along a linear array. But this area that we've described a lot over the last decade and a half using electrocorticography, we've now studied using single-unit recording. These are very, very rare cases where there's an ethical and medical access to this, but over the years we've had a couple. And what you're looking at is unpublished work where from a single neuropixel we can record, for example, 91 neurons. And this is what the neurons are doing in a task where people hear a word, wait, and then are given a prompt to speak the word. So they're basically just repeating it. And one of the things you'll see is that in the precentral gyrus, the motor cortex, you can see it's not just activated during speaking, but it's a subset of those cells are primarily responding when they're hearing the word. So this is now at the cellular level resolution, showing that there are these cellular subpopulations that are very much responsive when you hear a word, and then there's other populations that are there preparing to speak the word and then executing the word production itself. You can look at other parts of the precentral gyrus, you can see something similar. If you look at Broca's area, it doesn't look like much interesting going on there at all, which is fascinating. If you look in the auditory cortex, you can see a lot of responses to hearing the words and a couple that are during the production, but really strong responses to hearing the word. This is the cellular level. These are dozens of neurons that are recorded simultaneously just through one single column. It's really incredible technology. And if you look at this, over multiple data sets, we're now talking about single neuron at scale. So, for example, this is an example of a couple of data sets together, 500 units along these different phases across these different brain areas. And so we're now starting to enter this new phase of human neuroscience where we not only can do the single unit, but we can do it at scale. We can get a large population of neuron to understand more holistically how the population represents information. And one of the things that's super exciting about this is not just seeing the population, but the different kind of examples. So this is an example of neurons in the precentral gyrus. I've been describing how we've been looking at this coding. It's way more diverse when we look at this cellular level. So this one particular neuron is only active during listening to this one word, nobody. But here's another neuron that is activated when they say they in the speaking. This is another super interesting one where it's active during both listening and speaking, but for the same word. So it's activated when you hear a word in the sentence, and then it reactivates when you speak that word. We call that a mirror neuron. And then another set that are activated when people get the go cue. And then another set that are activated when they heard a word in the listen stays activated all the way until you actually say the word, like holding it in a kind of memory form. This is all happening within this cellular level population. So these are what we call mirror neurons. We can look at the timing of them and figure out what words these are encoding. Here they're responding to the sound of the word, and here they're involved in the production of the word. The same neuron. Here's an example of what we call bridge neurons. These are activated by hearing a word, and they stay activated until you say the word. Really, really interesting phenomenon, and probably has a lot of implications for understanding how we learn to speak, a lot of other phenomenon. This integration between how we hear and how we speak, all happening at a cellular level in the premotor cortex. So I just want to finish here and thank the people in my lab, but most importantly, thank all of you at NIH. It's been an amazing collaboration with the Institute, its leadership, the Brain Initiative, NIDCD. Thank you so much for enabling all of this work.
We're happy to take some questions. We have a few online. If there's nobody, please, up there.
Is this working? Okay.
Yes, it is.
Hi. Thank you for the talk. I have two questions, if that's okay. The first one is, like, when I'm trying to say something to somebody, but, like, I have passing thoughts that, like, I don't mean to vocalize, you know, like, oh, I'm really tired today or something like that. Or, like, when someone asks me how I'm doing, I'll say, like, oh, I'm doing well, but in my head, I might be like, oh, I'm really sleepy. So with these patients, since they're not trying to vocalize that thought, would it not detect it and would it not come out on the
That's
interface?
right. Yeah, and we've actually tried that. We try to decode what people are thinking about just to see if we could, and we can get above chance, but it's not great. I think for that you need to record from different brain areas and probably even more resolution.
Okay.
So this is not really about decoding the thought. It's really about decoding what someone is trying to vocalize.
Where would those thoughts be, like, stored in the brain?
That's $1 billion question, and probably actually throughout the brain, really distributed. There's not a lot of places that we can do surgery on that fundamentally changes someone's ability to think. So we think that this kind of information is very distributed.
Thank you. Second question is kind of similar, but the AI, like happiness, sadness, like expression that comes out.
Yeah.
I'm assuming that's also from a different brain region than the motor speech region. So how's that generated?
Actually, so the insight here is actually the opposite, which is that all of these things operate at the muscles of our face and orofacial structures. So our brain has one mode of moving those to create words and the sounds of words, but another mode is just for expression. And so we don't just use the vocal tract, of course, to speak. We use it for so many different things. So it's actually the same brain areas, but the coding of it within those is different for those gestures.
Okay, thank you very much.
Eddie, that was just a fantastic talk. So I have a lot of questions, but I'll only ask one. And the one thing that I think about when you talk about implanting people so long after they've spoken, you know, you would expect that there might be some degradation of the brain's ability to actually produce the neural activity because they haven't spoken. And I wonder if one thing that maintains that function is the last couple of slides that you showed about the integration of the hearing and the speech, because, you know, we think about cochlear implantation and you have cross-modal plasticity that really prevents people who've been long deafened from benefiting from that, but you don't see the same kind of thing here. So just wondering your thoughts about that.
I think that was thank you, Dr. Tucci. So, the question is, like, after not speaking for over a decade, is that part of the brain still working? And that was the biggest question in my mind before we started this, is that part of the brain become dormant? And it's really clear that that's not the case. Like you said, people hearing words, people understanding words. I didn't show this, but there's actually reading responses in this part of this area that we thought was just about motor control actually has a lot more sensory processing, including reading and listening. And I think that when people internally have inner speech, or even when they're trying to express themselves with a pointer and whatnot, they're still using their internal speech to generate words. It really goes beyond just the motor control. And so I think there's a lot of hope that those circuits are still functional, and that if we can tap into them at the right resolution, we can unlock, what expression is within them. Yeah.
Yeah. So my question was actually similar to what was just asked. I was curious if there is any sort of difference in the data that you get from people who are able to utilize their articulatory kinematics, like they're actually able to form the words, versus those that aren't. Is there any sort of is there more noise in those that aren't actually able to form the words with their mouth, or do you see anything like that?
Yeah, decoding from someone who's got severe paralysis is nowhere this close to someone who speaks fluently. And so while it's there, while it's latent, it's not as clear as those who are speaking every day, all the time. And so one of the things that we're very interested in, what we've seen anecdotally, is that this signaling actually changes over the weeks, days, and months that we do the training. The signals become clearer. The analysis becomes a little bit more straightforward. However, it's not just that. We see that people are even having increase in the muscle tone and their ability to move. They still can't speak, but it has this additional effect, like almost rehabilitative, because for the first time, they're trying to speak for hours per day. Now it's through a prosthetic, but we're seeing the byproducts actually on their actual oral facial musculature. Thanks for the question.
Give you two more. Go ahead.
Sure. Hi. My question was regarding the second patient, where they'd go ahead and read a question back, like the entire sentence. At the end, after it was decoded, it didn't generate like a question mark. Is that because the AI is having a difficulty with understanding like which words are being stressed at the end of a question?
Yeah.
Or,
Yes. So short answer is yes. We did not build in the stress. We are doing that now. And so the newer models can highlight words, that can capitalize words, that can stress words, that can put question mark, and convey some of that intonational information.
are you still recording from the same part of the brain, or
Yes.
are you still using like...
Yeah, so we're not tapping into like the semantic aspect or the meaning, that it's a question. We're really still tapping into how we express that in English, for example, by changing the pitch of the voice and translating that into stress or emphasis points. Yeah.
Thank you.
Yeah.
So in some of the talks that I heard about spinal transactions that get rehabilitated with different methods, there is actually an improvement in motor function that seems to be related to fibers regrowing in the spinal cord. And I was wondering if you see something similar here, where maybe the degree of paralysis diminishes over time, or any other effect. I mean, you may have already answered this question to the other person who asked from this microphone.
We have observed that strengthening of muscle tone and movement strength. I don't know if it's because of anything like new cells or, but probably synaptically, these circuits are being strengthened. In the setting of a brainstem stroke where the injury is fixed, I think that's where it's the clearest. With ALS, and we're studying this now, we are in this complicated regime where the paralysis is a degeneration, and at the same time, we're trying to get the decoder to work. And so it's a little bit of a moving target, and our current focus is really how to build decoders that can adapt for some of that degeneration over time. And it's not a trivial thing. It's actually quite important.
Thank you.
All right. Thank you so much again. That was absolutely wonderful. So everyone, please join us for a reception in the back here, and let's give Dr. Chang another round of applause.